Well, hello, everybody, and welcome to our virtual community or virtual myeloma workshop here at the International Myeloma Foundation. My name is Joseph Mikhael. I have the privilege of serving as a medical advisor to the IMF, and I am joined tonight by some stellar people that I'm going to introduce to you in just a moment. And tonight's topic is relapsed multiple myeloma. We're sad to say that, unfortunately, almost every patient with multiple myeloma at some point will relapse, meaning the disease will come back. But tonight, we're going to demystify that. We're going to talk about the options, and we're going to, in a very patient and care partner friendly manner, spend some time thinking about how we can tackle this relapse together. So it's a real privilege. I know there are several hundred with us tonight, and you may be listening to this after the event during the recording. We're welcome. We want you to feel welcome. We're very glad that you're with us tonight. I really think this is going to be a fantastic program. A few things. We really try not to turn this into Charlie Brown's teacher, where we just teach, nobody gets a chance to interact. So we do really encourage Q&A. We've scheduled Q&A. We love questions at the IMF. So you'll see in the window that is there that there's an opportunity for you to either send anonymously or by your name a question that you have for us. We're going to have two dedicated Q&As. I'll show you the agenda in just a moment. And if we don't get to your questions, as some of you know, I love answering questions, and it's what we do often in our YouTube videos. I hope you're subscribed to the YouTube channel for the International Myeloma Foundation. And I spend a lot of time in the studio answering these questions so that you can get your questions answered. We'll do our best to get to as many of them as possible. If you do have a technical issue, you can also, as you can see here, use that Q&A box to send a question to the support team. We have an incredible team here. They're fantastic. They get people up and running very quickly. We all have technical challenges here and there, so don't feel badly about it. We'll get you through it so that you can enjoy the rest of the webinar together. As I noted, this is not just a one-time deal. There is replay. And if you are with us tonight, you may not be able to watch the whole thing, or you just love it so much you want to hear Dr. Costa over and over again, then you can go to the website here, as you can see, and under the tab that's called Publications and Videos, you see all of our IMF videos, and you'll be able to pick up on our community workshop, as we're calling them here, our virtual community workshop. And then lastly, we want to hear from you. We want feedback. We've changed these workshops. Some of you may remember a couple of years ago, we used to try to cover everything if there was about myeloma into one evening. It was a little heavy. And now what we've done with our 3 virtual workshops is we have one focused on newly diagnosed multiple myeloma that we did a few months back. Tonight, we're going to be talking about relapse, and then later in the fall, we're going to be covering controversies in myeloma, some of the challenging topics that are difficult to navigate that we want to navigate with you. So we want your feedback. Is this the right way to do it? What else can we do? What other topics should we cover? Please do that. You'll see an opportunity to do that at the end. And we want to give a shout out to our sponsors that make these workshops possible: AbbVie, Amgen, Johnson & Johnson, Legend, Kite, GSK, Karyopharm, and Pfizer. We're very thankful for your partnership and for your support of these programs that we do together. On the subject of support, one of the things that the IMF focuses in on are support groups. And support groups are where so many wonderful things happen. This is where patients can and their partners gain education. This is where you can be empowered. This is where you can share experiences with others. This is really a marvelous opportunity in so many ways. Sometimes people hear support group and they think it's really where I go and cry. Not that tears aren't shed at times, of course, but it's so much more than that. And as you can see, we have support groups geographically by regions, and you go to that same website, myeloma.org. This map will come up and you'll be able to find a support group that is close to you, or just reach out to the SG team, support group team at myeloma.org. But we also have these shared experience groups that are not necessarily geographically oriented, but more about unique experiences, like patients who have young children, patients who do not have a care partner, veterans, those who are Spanish speaking. We have now a dedicated support group in Spanish. We have a support group that focuses on smoldering multiple myeloma, a support group that focuses on high-risk myeloma. You're going to hear a little bit about high-risk multiple myeloma tonight, and these others that you can see here. And these really provide unique experience in addition to the geographical support groups that you can take advantage of. Now, here is the real lineup. I always like to say I'm the appetizer. The main dish is who you see there afterwards. We have three extraordinary individuals today who are committed to the lives of their patients and their families. Dr. Luciano Costa comes to us from the University of Alabama. Shonali Midha comes to us from Dana-Farber Cancer Institute in Boston. And Tricia Mangan comes to us from the University of Pennsylvania. These are three centers, actually, that have been very sentinel to the development of many of these great therapies that we're going to be discussing in multiple myeloma. So for my three friends here, welcome. It's great to see your faces. Your faces are on the screen now, so everybody gets to see you live and on the photo, and we're just very, very thankful that you'll be able to participate tonight. Well, how is it going to happen tonight? This is what's going to happen. So after my introduction, I'm just going to try and level set us and make sure we understand what is relapse. How do we think about it? I have these, what I call the 10 principles or 10 key things you want to know, unfortunately, when your disease relapses, to empower you to make the right decision. Then I'm going to turn it over to Professor Costa, who's going to talk to us about CAR T-cell therapy. What is this whole notion of CAR T-cell therapy and how do we use it? What, when, and how? And then Dr. Midha is going to share with us the other two sort of newer modalities of treatment, bispecific antibodies and antibody drug conjugates. Also, what, when, and how. And then we're going to have some time for Q&A. We're going to have a quick break. And then Tricia is going to lead us in a discussion around supportive care. You know, Tricia, I always say it, I don't treat myeloma, I treat people. And so we want to look at the whole of the person as we care for them. And these new therapies, exciting and revolutionary, also have some unique side effects and some unique things that we want to make sure we understand to partner with our care team to give the best care possible. And then I'm going to pull out my crystal ball at the end and have a little bit of look-see into the future. Not that I'm a big fan of crystal balls, but it is amazing what is happening in myeloma right now and what is coming in myeloma. I've been around the block a little ways here. I won't comment on anyone else in the panel and how long we've been around, but 30 years in myeloma, and I have never seen a year like this with all the things that are happening. It just seems we have another FDA approval, which, by the way, I'm going to share with you a brand new FDA approval. It just happened a couple of weeks ago, not even 2 weeks ago. It seems that we have FDA approval after FDA approval, and that's great because it gives us more options. And that is really part of the focus tonight. We have reached a point where we have so many options. We really want to tailor the right option to the right patient at the right time. We'll have a little bit of Q&A at the end, and so we'll wrap up planning for this whole webinar to be approximately 2.5 hours. All right, as I like to say, all in favor, sit down. Motion carried. Let's dive into the first talk. So let's talk about what we think of as what is relapse, how do we approach it? And what I would like to do is talk a little bit, why does relapse occur? And then these important principles, 10 important principles that I want to share with you as you think about selecting the right therapy. As I mentioned before, we have so many options. I've joked before that it's kind of like the Cheesecake Factory menu. I don't know if you've ever been to the Cheesecake Factory. When my kids were younger, I think we used to go more than now. But I mean, you need like a table of contents for that thing. It's ridiculous, right? There's just so many choices. And I don't mean to belittle something so important, but I love that we have so many choices in multiple myeloma. We've just gained another choice, as I'll share with you in a moment. So it's really important that we think through how we're going to decide that, how we're going to walk through that menu. And a lot of that has to do with this shared decision-making process that Tricia is going to speak to us a little bit about as well. And then it really is important for you to know those options because we know the majority of our patients are treated in the community, and very often it's hard for a community oncologist who takes care of 40 different types of cancer to be updated in multiple myeloma. So we want to make sure you're updated. Some of you have seen some kind of slide like this. The nerdy hematologists here, we all talk about this slide on a regular basis. I actually think with time, this slide picture is going to change a little bit, the way we've changed the thinking of myeloma. But the concept of this slide is actually relatively simple. What it basically means is before someone develops myeloma, they have what we call a precursor condition, pre-myeloma. This is what we talked about at our first virtual workshop of the year when we talked about MGUS, or monoclonal gammopathy of undetermined significance. Smoldering myeloma, when patients have a little bit of that abnormal protein in their blood, but not enough to justify treatment, and some of them may never need treatment. But unfortunately, once someone crosses that threshold from so-called smoldering to active multiple myeloma, that's when we have to invoke treatment. Thankfully now, with the treatment options that we discussed at the last workshop, over 95% of people respond. which is really remarkable. I mean, Luciano, we can remember the days when that number was less than half of that, would have that kind of response to a first-line therapy. And here we are now with almost every patient responding. And thankfully, patients respond and typically actually respond now for several years. The sad reality is the disease tends to come back. Not only so, it tends to come back, and that's why the colors change on these little clusters of cells, is that they're what we call clonal evolution happens. You don't have to worry about the science of it, but it just basically means that the disease kind of mutates and changes and becomes more aggressive. And historically, as we started treating patients with each relapse, the disease kind of got more aggressive, right? It was just sort of a more and more difficult disease to treat. And that's why often by the time someone had 3 or 4 relapses, there was almost nothing that we could do. Thankfully, that is overall changing. Although the biology is similar in terms of how the disease can mutate, it really is improving with time. So it makes us wonder, why does someone relapse? We can treat people and we can almost see the disease disappear. And in some cases, we see what we call MRD negativity, which just means that when your myeloma is being tested, as you know, we typically test it in the blood. Some patients need it tested in the urine, where we look for that abnormal protein that are made by the bad cells in the marrow. And we don't always have to do marrow tests, of course, so that's why we can monitor the disease in the blood or in the urine. But when we clear it all in the blood and the urine, then we do a bone marrow test to see if there's any disease left. And sometimes we can look for what we call MRD, or minimal residual disease, is where we look for 1 in a million cells to still have multiple myeloma. And this has been a lot of Dr. Costa's work over a good part of his career, trying to understand how important that MRD negativity is. And so sometimes we can say to a patient, hey, we've done all the testing, your blood's clean, your urine is clean, and actually when we do this bone marrow test, we have no detectable multiple myeloma. We call that MRD negative. But the reality is there is probably still some disease left. And sometimes it's a tiny amount of disease, but sometimes, unfortunately, it is as I've often said, the last soldier to be killed is Rambo. And so sometimes the last little bit of disease can be very difficult to get rid of. And we think one of the reasons why patients relapse is that little bit that we thought was gone. You vacuum the floor, you think it's clean, but it probably is actually still some dirt there. And then that accumulates, and all of a sudden you've got relapse. The other reason why we think relapses occur is that the immune system, which is always trying to keep everything in order. Your plasma cells are wonderful cells. We need them. They make the antibodies, of course, that protect us from infection. But when someone has myeloma, there's a tendency for those plasma cells to kind of go off the rails and become cancerous. And so the immune system is trying to prevent that. But sometimes the immune system just can't. Just like a good teacher can't always control the students, right? Sometimes there's going to be a little bit of chaos in the room. And that's unfortunately what's happening. And so that's why as we talk about treatment tonight, we're going to be talking a lot about the immune system and the importance of the immune system to be restored and to allow patients to have that greater opportunity to stay in remission and not in relapse. Well, here is at least the first page of the Cheesecake Factory menu. I mean, if I don't have time tonight to walk through every one of these, belantamab, mafodotin, bortezomib, carfilzomib, CAR-T, daratumumab, elotuzumab, iberdomide, isatuximab, ixazomib, lenalidomide, pomalidomide, selinexor, T-cell engager. I mean, the list goes on, and you can see that literally it's been cut off a bit at the bottom. I think that's almost prophetic. And the idea is that there is just so much that's happening, and we're going to be talking through a lot of these tonight. I've put one in red because it was literally just approved. It's a new drug and a new class of drugs called iberdomide. The trade name is Zenbexis. I've not been given a very clear reason why it's called that. All I can say is when I say it, it makes me feel very zen, Zenbexis. But let me just quickly highlight this to you, because this is a new class of drugs that we call CELMoDs. If someone really wants to know what it stands for, it's a cereblon E3. That's how I remember it. E3 ligase modulator. That's a big worded way of saying this is a drug that very meticulously and carefully is able to grab onto something in the cell called cereblon. Cereblon is something that when we squeeze it, it triggers a pathway that helps destroy the cell. The drugs we use now in the class of IMiDs or immunomodulatory drugs, basically lenalidomide or Revlimid, pomalidomide or Pomalyst, and for those who may have had it in the past, thalidomide or Thalamid, they kind of like this little blue dot on the left here, they kind of pinch cereblon. They squeeze it a little bit. But these cell mods surround cereblon like a horseshoe, if not a full circle, and they squeeze it hard. And so that really becomes a more effective dosing. It's even a much lower dose. For example, typically with Revlimid, we use somewhere between 5 and 25 milligrams. For pomalidomide, we use 2 to 4 milligrams. Here, it's just 1 milligram. And so it's a very on-target drug that can help trigger this pathway to destroy the disease. And I don't have time to go into a lot of detail, but I'm going to quickly point out that this drug was just approved by the FDA in combination with daratumumab or Darzalex and dexamethasone based on this, what was called the Excalibur study. If you're into really cool names, you're getting a lot of them tonight. We have studies called Monumental, Majestic, Excalibur, Magnetism, a linker. I mean, I used to think when I was in medical school, only cardiologists had really cool names for their clinical trials, but that has clearly changed. And now we have pretty exciting names for our myeloma trials as well. But based on this Excalibur study, makes me sort of want to sit at the Knights of the Round Table. We saw that when we added iberdomide to daratumumab and dexamethasone, they really had a profound impact. And what they did in this trial is they compared it in a phase 3 trial to a common triplet that we use, DVD or daratumumab, Velcade, and Dex. So interestingly, it's a triplet versus a triplet. We don't often see that. We'll hear a little bit from Dr. Midha later about another set of studies with belantamab mafodotin that went triplet versus triplet. Often it's triplet versus doublet or 4 drugs versus 3 drugs. But here, 3-3 going head to head, it literally doubled the number of patients that got into that MRD negativity that I said. So that really speaks to the power of this compound. It also speaks on a separate topic, because I don't have enough time today to go over it. It really speaks to a remarkable reality that MRD negativity is now an endpoint that the FDA is using to grant approval for drugs. So you'll hear a lot more about Zembexis or iberdomide in the near future, but I wanted to give you that as a preview. Now, it looks like there's a lot on the page here, but I'm going to make this very quick because I really want to get, as I say, from the appetizer to the main dish and hear Dr. Costa. So there are 10 principles of treatment, and let's start with number 1, which is change the current mechanism of action. Typically, if I'm using a drug and the disease wakes up while on that drug or combination of drugs, we don't want to keep using it, because it means that the disease is smart enough to overcome it. And so it's time to make a change. So typically, if you're on a certain drug or a combination of drugs, your doctor is going to want to change that. And this is why we have so many classes of drugs. And this is one of the reasons why we record this video, because there's no way that I get to read all these off to you. There's 2 pages of this, and you can't get all the detail. I did put one in red here, which is one of the monoclonal antibodies we have called cilta-cel or isatuximab, just got approved. Not so much the drug was approved, it was already approved, but the way we deliver it. We went from giving it IV to now we give it through this thing called OBI or on-body injector, which is just a little device that kind of sits on the skin. As I say, as a good Canadian, it makes me think of a very small hockey puck, and it just sticks to the skin, and with a tiny needle, only 30 gauge, which is kind of like an insulin needle, it's so small, it delivers this drug over about 12 to 13 minutes. So it's not just that we're developing new drugs, we're developing new ways to give these new drugs. But we want to change that mechanism of action. And here's page 2 of we have all these new mechanisms of action, including that new cell mod that we just added to the bottom, the Zembexis or the iberdomide that I just described to you, as well as a whole series of things that are coming that I'm going to get to later. Principle number 2, don't save the best for last. We don't want to say, oh, let's wait until the disease has many more relapses before we use our best therapy. We have learned over time, it's always better to use the best therapies early on. Number 3, which we're going to hear a lot about tonight from our keynote speakers, we are increasing the use of CAR T-cell therapy, bispecific antibodies, and antibody drug conjugates. And we're going to explain what those are if you're not familiar with them later, but this is going to be very much discussed tonight, because this has really changed the way we treat it. If you go back just 10 years, doesn't it seem like a lifetime ago, Tricia, how we were treating myeloma 10 years ago compared to how we're doing it now? It really is amazing. We balance what is best for the patient, how we give it, and we'll talk through all of those options together. I said it earlier, I don't treat disease, I treat people. And so when we do treat people, we have to look at their whole context. We look at the patient themselves, we look at the disease, and we look at the treatment. And because we have so many options now, we try to mix and match. So it may be different if you have high-risk disease versus standard risk disease. It may be different if you have diabetes or other medical issues that might interfere with certain drugs. And then, of course, it very much depends on what you've had before. Those are the kinds of things that we decide. We also know that the depth of response matters. We don't want to give patients a drug that is not going to get a deep response, trying to, as much as we can, achieve that MRD negativity that we talked about earlier. Because as we get people into remission, we go from giving them what we call a partial response, where the drug has worked such that 50% of the disease is gone, to very good partial response, 90%, to complete response, where it's actually all gone. And the highest level of that is MRD negativity. Now, this is a very ugly slide, but it's actually beautiful to me, the myeloma nerd, because I love this concept here that combinations are really important. You know, we've really overcome much of HIV, not because we came with one magic pill, but because we found the right combination. In many respects, that's what we're doing with multiple myeloma. We're combining things. And so you're going to hear a lot tonight about drugs that target the outside of the cell and drugs that target the inside of the cell, all these weird numbers and letters that almost look like a different language. You know, we have friends of ours who spend their whole life hanging on to one little phrase there, and they study in their lab just the IL-6 pathway. I'm like, dude, you need more of a life. No, it's wonderful that they do that. We need our basic scientists to study these things, but it's putting it all together that's so beautiful in myeloma. So as we think about how we take down myeloma, like we want to take down any enemy, it's a very strategic plan. And tonight in particular, you're going to hear a lot about these drugs that target the outside of the cell, also the inside of the cell. Number 7, sequencing is important, but it's not fully known. You're going to hear about these different drugs, and someone's going to say, well, which one should I get first, Dr. Joe? Well, it depends, right? It very much depends. What are you going to choose on your menu tonight? Well, it kind of depends. Sometimes you're going to choose one over another, and in another situation, it may be very different. So we have learned a few lessons. Dr. Costa actually led the International Myeloma Working Group guidelines that we published not long ago, trying to say, okay, if you had everything available, generally speaking, we might favor CAR T first. That's why we put CAR T first in the discussion tonight. But as he's going to admit to us later, some of the data coming from bispecifics is challenging that notion. But again, we want to be able to use the best now, but also not burn bridges for the future. And we also learned that a lot of these drugs work by virtue of the T cell. Dr. Costa is going to explain to you this in just a moment. But as a preview, think of T cells as soldier cells that live in your body, that have the ability to be trained and attack your own multiple myeloma. And we know that as we use these treatments, we engage T cells. We want to make sure those T cells get a rest between the treatments that we give. Number 8, always include a quality of life discussion. This is so fundamentally important because we don't treat a bunch of plasma cells, as we said, we treat people. And so making sure you know what this means for your quality of life is absolutely critical. Trisha's going to share more of it later, but we believe strongly, I actually think the Nurse Leadership Board at the International Myeloma Foundation have really been the world's greatest proponents of this concept of shared decision-making, which just basically means that the doctor doesn't just come in and say, you'll take it and you'll like it, right? It's much more about saying, look at all the options. What are your preferences? And now let's mesh those together and let's decide together what is best for you as the patient and their partner and decide where to go from there. We're going to hear more about that great model a little bit later. And then lastly, number 10, and this is for no self-promotion. This is for your benefit. One of the things that the IMF has is an info line that you can call into and ask, and we have a network of experts. We believe that in the day in which we live, considering at least an expert opinion is a good idea. I prefer to use the word expert opinion instead of second opinion. Second makes it sound like the first one was wrong, and more often than not, the first one wasn't wrong. It may just need a little bit of editing or a little bit of support. But this is where we have people who really like the team with us tonight. Tricia, Shonali, and Luciano all spend most of their life immersed in multiple myeloma, and being able to see someone like that can be particularly helpful to you. And with that, I come to the ugliest yet most beautiful slide of all, the slide that I'm running out of space on because I keep having to add to it. It is amazing, the list of things that we have available to us and what's coming. Myeloma has really changed. And hopefully tonight you're going to learn more about how we approach that relapse. So with that, I will say thank you for listening. We are right on time, Sir Luciano, and we are going to turn it over to you, brother, to talk to us about CAR T-cell therapy, what, when, and how. So Professor Costa, coming to us live from the airport in Sao Paulo, Brazil. Thank you for joining us, brother. The floor is yours.
Thanks, Dr. Joe, and thanks the International Myeloma Foundation for this a great opportunity to talk to you tonight about CAR T-cell therapy, what, when, and how. Some of my disclosures. So, the term, I like to, start by kind of deconstructing the term CAR T-cell therapy. And that means chimeric antigen receptor, T-cell. And many of you might be aware, some of you might not, but the term chimera comes from this mythological figure, which was an animal with three different heads. It's the body of a lion with the side of a goat and a tail that is a snake. So we incorporate that into medicine to explain when there is the coexistence, so to speak, of pieces of two different organisms on the same being. And I'm going to tell you why we use that term to designate CAR T-cell. And to understand that, you have to understand what a normal T-cell is. When we were in medical school, for some of us a few decades ago, we learned that T-cells are part of our innate immune system. So we all have those cells in our body, which is a way I'd like to explain to my patients is a natural way to defend against infection. So for example, if you acquire hepatitis, let's say hepatitis A, that cell infects your liver cells. And the virus go inside the liver cells, and they are not visible to most of your immune system. But then your liver cells start making proteins that are unique to that hepatitis virus that is not part of your normal being. And our body, amazingly, has a large repertoire of T cells with different receptors, and somewhere in our body, there is a T cell that is capable of recognizing that foreign antigen through this very meticulous interaction between the T cell receptor and something called MHC class 1 that shows the antigen from the cell to the T cell, and the T cell goes and destroys that cell that was infected by the virus. Well, that doesn't work very well against cancer, or in most cases, because the cancer is a corruption of a normal cell, right? So every single protein in our myeloma cell is a cell that our body considers a self. So we don't have, swimming around our blood, T cells that can recognize our myeloma. Otherwise, we wouldn't have myeloma in the first place. So what is CAR T-cell? It's essentially deploying this ability of the T cells to kill other cells against myeloma. But in order to do that, we have to do some engineering of the T cells. We essentially have to collect these cells from the patient and insert into that cell a gene that now makes the T cell make a receptor that recognizes a protein on the myeloma cell. So regardless what the T cell's original job was, that is put aside. Suddenly the T cell receptor no longer matters, and that T cell now has been repurposed through a chimeric gene, from a gene that comes from an outside organism, to recognize a protein on the surface of the myeloma cell. And that's why it's called chimeric T cell. So this process is a little bit complicated. And what I'm describing here to you is the process for autologous or self T cells. So T cells made from your own blood cells. It always starts with leukapheresis. Essentially, is a process where we make your blood go through a machine that separates the white cells in your blood and admix with those white cells that are those T cell lymphocytes. Then those T cells are sent to a factory, to a manufacturing facility, where the T cells are separated from the rest of the blood, and they are modified with the insertion of that gene. And that modification is more often through the introduction of the gene via virus. So it's kind of fascinating to think that we have to infect the patient's cells with a virus, usually a lentivirus, that it serves as a vector that carries into the cell that gene that makes the receptor. And then there's several steps of growing those cells in the Petri dish, purifying those cells, and going through a system of quality control. So in the end of this process, which usually take a couple of weeks, we have a uniform batch of cells. Each one of them has the receptor that recognize a protein in the myeloma cell. And those cells are unique to the donor, is unique to the patient who gave the cells in first place. And those cells are ultimately infused into the patient in order for to combat the myeloma. Once they get infused to the patient, they multiply, they grow in number, they expand. And each cell that originates from the batch of cells is able to track the myeloma cell, engage with the myeloma cell, and kill the myeloma cell like a good T cell is capable of doing. Now, that receptor is not human, right? Because we don't make antibodies that can recognize self proteins. So we don't make antibodies that can recognize any target on the T cells. So we had to borrow those genes from other species. And we have now 2 CAR T cells that are available. One of them is called Abecma or ide-cel. That was the first CAR T cell approved for multiple myeloma, and it now can be used in patients with 2 or more prior lines of therapy, so long they receive therapy with a proteasome inhibitor like Velcade, an immunomodulatory agent like Revlimid, an anti-CD38 monoclonal antibody like Darzalex or like Sarclisa. And that antibody piece that goes with the receptor is made from mouse. And you're going to find this cute that the cilta-cel which is also known as Carvykti, is now approved for patients with lenalidomide or Revlimid-resistant myeloma who had 1 prior line of therapy. And that is made of llama. Now, there are some differences between them. Most of the molecule is similar. The Carvykti has 2 sites of binding to the protein on the myeloma cell that's called BCMA, while the ide-cel has 1 site of binding, which might be some reasoning why they have different efficacy and different side effect profile. Now, I'm just going to take you back before we talk about CAR T cell, and let's see how they become first available and why. So it hasn't been long, maybe 7, 8 years ago. You know, if Dr. Joe or Dr. Midha or myself had a patient in our clinic who had myeloma, had received, Velcade, Revlimid, and Darzalex, let's say, 1 of each class, and the disease was getting worse despite being seen out of these therapies, we could not expect much. Those patients would have a median disease control of about 4 months and a median survival of less than 12 months. So throughout the evening, we're going to show you several curves like this. And if you're looking at this for the first time, the way you read those curves is on the vertical is the proportional percentage of patients, and on the horizontal is the number of months, right? So if you have a perfect treatment, you have a straight line all the way to the top. Everybody stays alive over time. And if you have a bad treatment, you have a sharp drop because people start dying from their disease, right? So the more horizontal this line is, the better it is. And here you can see near half the patients had died only 12 months after starting a new treatment that for myeloma that had seen all three main classes of therapy. Then the first CAR T became available. That's ide-cel or abecma, and became available as the first approved CAR T cell in the United States, thanks to this study, where now instead of only 1 in 3 patients responding, we have more than 80% of the patients responding to treatment. Instead of responses lasting about 4 months, which was the usual for any therapy that we would use in the past, now we have disease responding for 12 months, and people surviving, much longer. People who had complete response on this therapy had a survival that was superior to 20 months. So head and shoulders above what we used to have up to that point. So this trial, this CAR T cell was compared to the regimens that we used to use until a few years ago for patients who had all the 3 classes of therapy. And you see here the CAR T is the line on the top, the blue, and the conventional treatment is the line on the bottom, the red line. And you can see clearly the CAR T comes on top. Patients receiving this therapy had 13.8 months of median progression-free survival, so the time they stay well without progression, versus only 4.4 months with conventional treatment. So this led to the approval of CAR T even earlier on patients who had only 2 lines of therapy. Now, the other CAR T, the Llama one, is CARVYKTI. And this has triggered people's imagination even more because of results like this. This is what's called CARTITUDE-1. This is the trial that led to the approval of this CAR T in first place. Again, patients with a median of 5 prior lines of therapy had been exposed to all the 3 main classes of agents. And now, instead of having survival being less than a year, we have survival reaching 5 years. So quintuple what we used to expect for therapy for patients in that circumstance. But what really trickle your imagination is it's now with 5-year follow-up, 1 in every 3 patients who used to have a disease where you couldn't expect to live more than a year are alive without any detectable myeloma cell in their body, which really brought up into the myeloma conversation the perspective that some of those patients might actually be cured and never see the recurrence of the myeloma. Time will tell. Of course, anything that works in disease that is so advanced is going to work better in earlier lines of treatment. So this treatment, cilta-cel, was also compared with conventional treatments, with treatment that we would otherwise use in patients with 1 to 3 lines of therapy. And what we see here is a much more common rate of response, close to 80% of patients achieving complete response when the myeloma protein is not detectable in the blood or in the urine, and the myeloma cells are not detected in the bone marrow. And 2.5 years post-treatment, virtually 2 in every 3 patients are alive without signs of the disease. So there's a lot of hopes that we're gonna, be curing some of those patients. And this, I like this graph because it shows you the same. Show you this principle that we're giving everybody the same therapy, a standard dose of this CAR T called cilta-cel. And you see patients with 4 or more prior lines of therapy, patients with 3 lines of therapy, patients with 2 lines of therapy, and patients with 1 line of therapy. So you can see the curve is closer to a straight line the earlier the treatment is used, really conveying the notion that this therapy performs far better in earlier lines of treatment. But you can tell, well, but Luciano, ever treatment works better earlier, which is true. But here it's a little bit more than that. Not only have disease that is less resistant, but we know now your immune system is better suited for this treatment earlier on, as opposed to after you have received multiple lines of therapy that have, in a way, eroded the quality of the T cells that are ultimately the manufacturing material to produce your own CAR T cells. Now, let's think about the logistics. That's the how part, right? And that's why this treatment can be a little bit complex and intimidating to some patients and to some physicians. Let's say, you are a patient with myeloma and you're seeing, your cancer doctor in your hometown, and the cancer doctor say, well, I think CAR T, it's time for you to go ahead and plan for some CAR T treatment. But unless you are being cared in a large center where they perform CAR T cell already, you have to be referred to a cell therapy center where you're going to see a physician who's a specialist in myeloma and a specialist in this therapy, who's going to, meet with you and see if the treatment is right for you. If the treatment is right for you, we're going to schedule apheresis. So that is the process where it will connect you to a machine that essentially centrifuge your blood, remove the part with the T cells, give the rest of the blood back to you. That's a one-time outpatient procedure, and some patients require a catheter placement to allow the right flow to go back and forth from that machine. Now, from that point onward, those cells are sent to a facility where it's being manufactured. So that's the starting of those 4 to 6 weeks to have, a CAR T cell product. Now, this treatment is being recommended to you because your myeloma is moving, is active, is progressing. And we do not trust myeloma to stay quiet while we produce those CAR T cells because it can be a little bit unpredictable. So we almost always employ what we call bridging therapy. And there's nothing magic or unique about bridging therapy. It's essentially any good myeloma therapy that it still makes sense in your case. Usually drugs that your myeloma have not seen before that are likely to work. And the goal of that is keeping the myeloma from making sick while you wait for those CAR T cells to be produced, and also reduce how much how much myeloma you have in your body, because the more you have, the more likely you are to have some of the side effects that we're going to discuss. Now, 4 weeks have passed or 5 weeks have passed, and now we know that your cells have been successfully manufactured. So we have a bag of, Abecma or Carvykti with your name on. We're ready for treatment. Before we do that treatment, there is some lymphodepletion therapy. So that's a therapy that is standardized, is 3 days of a chemotherapy that aims not at killing myeloma, but at killing some of the normal lymphocytes in your body. And the way I explain to my patient is, you want to kill the normal lymphocytes in your body. So now your body wants to make more lymphocytes. So your body starts producing the substances, the ingredients to make new lymphocytes. And right when your body is doing that, we infuse those CAR Ts. So they're going to find an environment that is very permissive for them to grow, to expand, and to multiply, because your body is producing all the juices, all the ingredients to incentivize, to encourage those T cells to proliferate. And then comes what sometimes is the harder part, which is the follow-up to assess the response that your myeloma had to this treatment and deal with some of the side effects. So as you can see, it can be a lengthy process. Sometimes we use the term, vein to vein. That is the time when you collect the cells until the time you infuse the cells. And best case scenario, that's about 6 weeks. And sometimes we use the term brain to vein, which is the time when you decide or you recommend transplant to that patient until the time that patient get infused, which can be 8 weeks and in some cases much longer. Now, I talked to you about bridging therapy, and that's a term you often hear. But we also have the term kind of holding therapy. Sometimes your myeloma start progressing. Your doctor says, well, I think CAR-T is going to be right for you. But until you can go and see Dr. Joe and have that plan, let me start you on some treatment here to keep your myeloma from getting too bad, right? And we use the term holding because it's trying to hold your myeloma down until we go on this treatment. And that's very important because some drugs we do not want to use as holding therapy because they can damage those T cells. They can keep you from yielding some healthy T cells for manufacturing. So it's not up to you, but it's up to your doctor to know what those drugs are. When you go to bridging therapy, we're already collecting the cells. So there's some more freedom. There's some agents, some drugs that can be used because we don't care if they damage the T cells anymore because the T cells have been already collected. And then there's the lymphodepletion chemotherapy that you're going to receive at the CAR-T center 3 to 6 days before having the infusion of the CAR-T. And that is very standardized. Almost invariably is a combination of 2 drugs called cyclophosphamide and fludarabine. So those are all chemo that you get before CAR-T, but they have different, they have different position, different sequence, and they serve different purposes. Now, and that's essentially what I discussed with you. We want with that bridging therapy to keep your disease from making you too sick before you get the CAR-T, reduce the burden of myeloma. And again, there are some drugs that we consider quote unquote prohibited before we collect the cells. For example, a drug called bendamustine, or sometimes some of the bispecific antibodies that Dr. Midha is going to tell us about. They don't go well right before CAR-T because they affect the health of the T cells. But they can be used after you collect cells for treatment. Now, you're going to hear a lot about sequencing treatment, right? We have all those new options, and some of that is some of our tasks and some of our challenges is how to use those treatments in a way that makes sense, that we don't burn bridges. And because a lot of the treatments Dr. Midha is going to tell us later, the bispecific T-cell engagers or the antibody drug conjugate, they go after the same target, which is BCMA, right? So it is expected that if you use those drugs before T, the efficacy of the CAR-T is going to be somewhat compromised. So if you give, for example, cilta-cel, we like to quote to the patients, the chances of response are about 95%. Well, if you have a bispecific T-cell engager, if you have a antibody drug conjugate against BCMA, that chance of response drops to about 60%. But more important than that, as you can see on the panel on the right, the duration of the response is much shorter. And it's from this reason, we have the recommendation that Dr. Joe mentioned, that if you have a patient in front of you who's a candidate for all those therapies, have access to all those therapies, we like to use the CAR-T first because, if you use the CAR-T, patient has a response, and later you have to use, for example, bispecific against the same target, you still have a reasonable chance of response, which is not 30, 40% down, it's more like 10% down, right? You have a lesser of a compromise on the bispecific after CAR-T than the CAR-T after the bispecific. And the other reason is we now have bispecific against a different target that is not BCMA, as Dr. Midha is going to show you. And one of those, or the only one currently available, is called talquetamab, that targets protein called GPRC5D. And we have some pretty good results from clinical trials and from real world showing that after a BCMA CAR-T, talquetamab works almost the same as if the patient had never had any immunotherapy, right? So you preserve that option for the future. You do not compromise the efficacy of that therapy by using CAR-T. Now, CAR-T is an extremely powerful, perhaps the most powerful anti-myeloma therapy that we have. However, it is not free of side effects. And some of the things that we deal with are something called cytokine release syndrome. And the way I explain this to my patients is, if you ever had a very bad flu that you stay in bed for a couple of days, feeling, with fever and chills and not feeling well, what is causing you to not feel well is your immune system responding to the viral infection and producing a lot of substances to combat the virus that we call cytokines. They are the coding, the signaling that goes between the different parts of the immune system to combat that infection. Well, here you have the same thing. You have a very pronounced and acute and extreme activation of your immune system, not to fight the flu, but to fight the cancer. So you're going to have a fever, most people do, and in more extreme cases, you can have a fever with low blood pressure or a fever and problems breathing. Now, that is called cytokine release syndrome. Most patients will have some, and nowadays we feel very comfortable about handling that side effect. It's usually in the first 2 weeks after CAR T. It's not something you're going to have a month later. We have great ways to mitigate that side effect. The other side effect is a bit more concerning. It's called ICANS, which is called immune-effector cell-associated neurotoxicity syndrome. Long words. But essentially, it's believed to be the effect of some of the same cytokines affecting your mind for a little bit. Can cause people to be confused, not being able to name objects, sometimes not be able to speak. In extreme cases, people can become irresponsible for a few hours or a few days. It can be very scary if you are, the patient or the family member. But again, is uncommon, less than 10% of the patients. The very bad case where you're not talking is probably less than 5%, responds very quickly to steroids, and hardly ever is fatal. So again, it's something that can be scary. We talk to patients about it, but it's not something that we who have experience in this therapy are particularly concerned about. And I find when I talk to my patients, what really influence their decision-making are the things that are far less common, also less predictable, and not always get better. So there are some neurologic side effects that can take place later. For example, cranial nerve palsy, when you lose the movement in one side of your face, or Parkinson-like syndrome. We don't fully understand why it happened or how it happens. Some of that can be reversible. Some of that may not. Fortunately, is uncommon. We talk about 1% to 2% of the patients. But for some people, when they hear 1% to 2%, that can become, important on their decision-making. And other issues are less of a concern. You have a risk of infection, and there's several things that we do nowadays that really reduce the risk or keep people from having life-threatening infections, and some issues with low blood counts. All those problems are getting better as we gain experience with those treatments, and most important, as we use those therapy earlier. It used to be that when CAR T became available, most of our patients had 6, 7 lines of therapy. They're being treated with their bone marrow full of myeloma. Nowadays, the majority of the patients that we're treating are being treated as a second or third line after responding to an effective bridging therapy. So we're seeing, those side effects getting better really in front of our eyes. So the current challenge that we have is the access. Is not a therapy you can get in every oncology office. It really requires a specialized cellular therapy center. Fortunately, there are hundreds in the United States that are approved and capable of doing this treatment. But, I'm in Brazil today, and I was talking to my colleagues here about this, and we only have a few sites. And in the whole country, they have done 100 treatments total. I think in the United States, we do 100 treatments every week when you count all the sites. It's costly. It's not a cheap treatment. And I think the timeline can also be a challenge. As I said, if you live 3 hours, 4 hours from a treatment center, sometimes our centers are overwhelmed, and we don't have, we don't have a slot for apheresis, or problems with manufacturing. And also we have some room for improvement in efficacy and the safety. Now, what is being done to improve? I think Dr. Joe is going to tell you a little bit about that. But one of those possibilities is what's called allogeneic CAR T. So now pretend you need a CAR T cell treatment, but instead of going through all the problems of being evaluated, have apheresis, be sent to manufacturing, wait 6 weeks, there is already a batch of T cells ready for you sitting on a shelf. And that is becoming a reality using donors and some genetic modification of the cells that you can use those cells to infuse in just about any patient without having to produce your own. That cuts the time from brain to vein from 8 weeks or more to as little as 2 weeks. So there's a lot of allogeneic CAR Ts being developed that might come to the clinic. And I think Dr. Joe is going to tell you later about what perhaps is the most exciting version of this, which is called in vivo CAR T, which cuts through all the chains. Essentially, instead of, we don't even have to produce cells to infuse on the patient. We infuse already the transgene, often in the form of a virus, wrapped by a lipid nanoparticles, essentially a fat envelope. And those that envelope tracks to the T cells that are doing their job, swimming around your bloodstream, enter that T cell and transform the T cell into CAR T. So your own body, within a matter of hours, is producing CAR Ts that are able to multiply, engage the cancer, and kill the cancer. That's a fascinating new wave of innovation that is coming to this field. And with that, I'd like to, again, thank you for your attention, and I look forward to the part where we answer your questions. Thank you.
Wonderful. Thank you so much, Dr. Costa. Wow, lots of great information there. And you're going to hear more about those side effects, a little bit more of those futuristic things in a little bit. But I don't want to take any more time for Dr. Midha. We're so grateful that Shonali's been able to join us tonight. Talk to us about bispecifics and antibody drug conjugates. The floor is yours.
Thank you so much, Dr. Mikhael. And that was an incredibly elegant presentation, Dr. Costa. I'm thrilled to join you guys today. And I'll get to focus on our off-the-shelf therapies, the bispecific antibodies and antibody drug conjugates in relapsed refractory multiple myeloma. These are my disclosures. And so as Dr. Mikhael has shown you, and others I'm sure have shown similar diagrams in the past, the treatment landscape in myeloma has evolved greatly. And so what I really want to focus on here today is this last row that looks at the approval of our first antibody drug conjugate, belantamab mafodotin, back in 2020 with somewhat of a storied history. It lost its, or GSK did not apply for its full approval upon the release of the DREAM-3 study. And so it was no longer available in 2020, late 2022 onwards. However, at that time, the bispecific antibodies came on the scene, with the first one being teclistamab back in October of 2022, followed soon by other bispecific antibodies, talquetamab, elranatamab, and most recently, linvoseltamab. The caveat in this being belantamab came back on the scene with one of its phase 3 studies that we'll talk about compared to a triplet regimen, and then led to, again, its approval back in 2025. And so it's a really exciting time, as others have mentioned, in multiple myeloma to see such incredible growth over time. But what does this translate to in terms of treatment? And so that's where we'll focus on the options available. And so we'll start with, what are bispecific antibodies and antibody drug conjugates? Bispecific antibodies are off-the-shelf treatments that physically bring a T cell and your myeloma cell in proximity to each other. So as you can see here, antibodies typically have this Y-shaped structure. And on one arm of it, you can think of it that it has a binding domain to a protein on the surface of the myeloma cell, which Dr. Mikhael had shown in the diagram previously. And on the other arm actually has a binding domain to a CD3 receptor on T cells. And by bringing these two cells in proximity to each other, that allows for the activation of the T cell, which can then kill the myeloma cell. The advantages of bispecific antibodies, of many that we'll discuss, include that they're readily accessible, especially for those patients that may not have that luxury of time. As Dr. Costa mentioned, there is a brain to vein and a vein to vein wait time involved in CAR T cell therapy, from the time of consult to apheresis and manufacturing of the CAR T cell product prior to its infusion into the patient. The other advantage here is this therapy is an immune therapy. It itself does not involve chemotherapy and does not, in most cases, require chemotherapy prior to its administration. Now, antibody drug conjugates are also antibody drugs, but a little bit different in that the antibody carries a chemotherapy payload. So as many of you know, daratumumab and isatuximab are monoclonal antibodies that we use often in newly diagnosed multiple myeloma. Similarly, antibody drug conjugates do harness the immune system to directly attack the myeloma through the stem of that Y portion of the antibody that binds to sort of the soldiers of your immune system, the macrophages and NK cells, that are then activated by seeing that antibody drug conjugate bind to the myeloma. In addition to that, when antibody drug conjugates bind to the myeloma cell, they're also internalized into the cell, and it's broken down to allow the release of this chemotherapy payload. And that then leads to direct blocking of the cell division pathways. And that is how antibody drug conjugates sort of work as a Trojan horse in allowing the chemotherapy payload to be released into the myeloma cell. And similarly to bispecific agents are off-the-shelf products, again, for patients that may not have the luxury of time for CAR T cell therapy, apheresis, and manufacturing. Now, why don't we dive into the details of these therapies? I'll start with the bispecific antibodies. As I mentioned, there are 4 currently that are FDA approved and available on the market. 3 of them that target BCMA, the same target that is utilized for currently available CAR T cell therapies, and 1 that targets a different target, GPRC5D. The majority of these therapies are given either as a subcutaneous injection, as you can see here, or IV. And they start as step-up dosing to prevent certain side effects, particularly the cytokine release syndrome that Dr. Costa mentioned, that we see with CAR T cell therapy, that we also see with bispecific antibodies. By allowing for a step-up process, that decreases the risk of cytokine release syndrome during those step-up dosing. Patients then continue this therapy either weekly or, if they're seeing a response, at a lesser frequency as time goes on. So unlike CAR T cell therapy, as we learned about, which in some cases we think of as a one-and-done approach, but that might be too oversimplified. That being said, bispecific antibodies are therapies that were studied to continue until progression. In terms of the T cell target across the board, that is CD3. These therapies are currently FDA approved in patients that have had 4 or more prior lines of therapy. However, we will talk about a little bit later, the caveat here being teclistamab in combination with daratumumab has received an approval for an earlier line of therapy as well. Now, how do these therapies gain approval? And so this is a summary of the response rates seen for each of these bispecific antibodies as single agents in relapsed refractory disease. So across each of their registrational trials that led to their FDA approvals, these drugs were studied as a single agent in patients with relapsed refractory disease or disease that has progressed, in most cases on a median of 4 prior lines. And as you can see here, whether it was teclistamab, elranatamab, linvoseltamab, all of our BCMA binding bispecific antibodies, or talquetamab, our GPRC5D binding bispecific antibody, response rates are impressively above 60% across the board as a single agent. The other point to note is many of these responses are deep, meaning patients are achieving a very good partial or even a complete response. This is another way to look at that. As you can see across these studies that included, in most cases, over 100 patients with up to 2 to almost 3 years of follow-up. Again, response rates above 60% across the board. Many of those, a third or more, are oftentimes complete response rates. And in those evaluable of patients that had MRD status that could be studied, nearly 90% of patients across the board were MRD negative at the time of assessment. Again, these responses are durable. So we're seeing progression-free survival of a year or above and median overall survival even beyond that, in some cases not reached. Now, these are not without side effects. Even though we think of chemotherapy as having a lot of side effects, as many of you know, immune therapy also has its own host of side effects. Oftentimes what we see with the bispecific antibodies are hematologic side effects, which oftentimes are low blood counts, anemia, low platelet counts, a low white blood cell count, or otherwise referred to as leukopenia or neutropenia, which can increase the risk of infections. As we mentioned with the bispecific antibodies, similar to CAR T-cell therapy, because these are all T-cell engaging therapies, we often also do see similar side effect profiles. So cytokine release syndrome can be seen most commonly during the step-up dosing for each of these bispecific antibodies, and hence the use of the step-up dosing. That being said, oftentimes the cytokine release syndrome experience, as you can see here, is low grade, meaning grade 1 or grade 2, which in the case of cytokine release syndrome means if patients did not require, in most cases, ICU support or support for a low blood pressure or significant oxygen support. Again, we see in terms of time of onset and the median duration of the cytokine release syndrome across several bispecific antibodies, this tends to happen during the first couple days during the step-up dosing, during the first cycle, and only lasts for anywhere from 15 to 24 or 48 hours. Now, to a lesser extent, sometimes we do see neurotoxicity as well, oftentimes during the step-up dosing, again, for these bispecific antibodies, to a lesser extent, often than what we see with CAR T-cell therapy, and often to a lesser grade, is in most cases can be resolved with steroids or supportive measures. There are slight differences from one therapy to the next. However, in most cases, we do not see significant long-lasting neurotoxicity. This, again, tends to be during the step-up phase of the dosing. Now, there are some adverse events that we do like to mention specifically because 3 out of the 4 bispecific antibodies bind to BCMA, which is highly expressed on malignant myeloma cells, however, can be expressed on precursor cells and B cells. Oftentimes, we do see an increased risk of infection. And this is something that we very commonly see, unfortunately, with BCMA targeting bispecific antibodies. And of note, many of those infections, in some cases nearly half, can be high grade, meaning they require IV antibiotics or hospitalization or additional measures to treat. Most commonly, these are respiratory infections or pneumonia. and or include upper respiratory viral infections such as COVID, influenza, or other opportunistic infections. And so Dr. Costa mentioned this before. There are several studies now looking at how do we combat this, most commonly with the use of prophylactic antibiotics, antivirals, and IVIG support to help limit the degree of infections that are seen. There are several ongoing studies as well to better understand which patient population may be at higher risk of certain opportunistic infections and how we can best prevent and prophylax for that in the future. With talquetamab being a GPRC5D binding bispecific, we do tend to see infections, however, to a lesser extent and to a lesser degree than what we see with the BCMA binding bispecific. That being said, because talquetamab binds to GPRC5D, it has its own unique set of toxicities that we do not tend to see with the BCMA binding bispecifics. GPRC5D is a target that was identified in multiple myeloma. However, it happens to also be located on certain keratinized tissues. And so in many patients, we do see skin, hair, nail, and oral or taste change as a potential side effect to talquetamab. Most commonly, we see what we call dysgeusia or taste change, which is seen in nearly 3/4 of patients on this therapy and is managed oftentimes with supportive measures such as good oral hygiene or oral rinses, saliva substitutes, and or oral steroid rinses if significant. However, it does require a multidisciplinary approach, as many patients do also lose weight in this regard due to loss of taste and loss of appetite. And that is something that we have to watch very carefully. In addition, we also sometimes see rashes, skin peeling, skin-related toxicities, nail changes to creating brittle nails or nails falling off, oftentimes which we manage with vitamin E-based nail hardeners or creams. And again, the skin changes managed with frequent moisturizing, as well as other potential therapies to use to treat skin changes and or rashes. We do see that by decreasing the frequency of dosing, that can decrease the experience of these toxicities, as well as managing with supportive measures, as I mentioned. Now, how best do we get the most out of our bispecific antibodies? As I mentioned, the combination of teclistamab and daratumumab was recently FDA approved by accelerated approval based off of a Majestic 9 study. And so in many cases, we see that by combining bispecific antibodies, as Dr. Mikhael mentioned earlier, with other therapies that may target intracellular targets as opposed to extracellular targets, so targets on top of the myeloma cell versus inside or supportive cell in the environment around the myeloma cell, can induce improved outcomes. And so what was published late last year, as you can see here, why the survival curve on the right was improved outcomes in patients that received the combination of teclistamab and daratumumab in patients that had 1 to 3 prior lines compared to standard of care. In this case, the standard of care arms included daratumumab, pomalidomide and dex, or daratumumab, Velcade and dex. More patients achieved a response, nearly 90%, with nearly 60% achieving an MRD-negative response, so very deep responses, with a median progression-free survival that has not yet been reached, as compared to those on the comparator arm, the control arm, in which the median progression-free survival, so time until myeloma started to progress, at 18 months. Now, one thing of note, we obviously did see an increased risk of infections with the teclistamab containing or the BCMA bispecific containing arm. that was comparable to those in the control arm of daratumumab pomalidomide and dex, or daratumumab Velcade and dex. Patients were provided with antiviral and antimicrobial prophylaxis and IVIG. Similarly, combinations were studied with talquetamab, among other bispecific agents, and these were results from the Monumental 3 study presented this year that looked at talquetamab with daratumumab, or talquetamab with daratumumab and pomalidomide, compared to daratumumab and pomalidomide with dexamethasone. And again, we're seeing response rates that approach 90%, nearly 90%, with many of those responses being complete responses or very deep responses. And again, seeing improvements in survival, in both progression-free survival with talquetamab, daratumumab and pomalidomide, or talquetamab and daratumumab. So beyond combining bispecifics with other myeloma agents, you can also combine bispecifics with themselves. And so this led to a number of studies, but this is one example of that, the Redirect 1 study that looked at the combination of teclistamab and talquetamab, so utilizing two separate myeloma tumor antigens, one targeting BCMA and the other targeting GPRC5D, in which, again, we're seeing very high levels of response in patients with relapsed refractory disease. And particularly what I wanted to point out here is the responses in patients with extramedullary disease, which we recognize as a high-risk factor in multiple myeloma, and sometimes leaves patients with very few options when we see disease outside of bone. And what was encouraging to see here is across several dose levels, the response rate in patients with extramedullary disease was, again, nearly 70%. And in those at the recommended phase 2 doses, we saw response rates approaching 86%. So that was very encouraging to see in a very hard to treat population and disease subtype. Now, as I mentioned, the T-cell engaging therapies are not without their issues. So what we are still learning is how do we best sequence? Dr. Costa touched on this in terms of sequencing CAR T-cell therapies and bispecific agents. What do you do if you can't get one or the other first? Can we use CAR T therapies first, or should we use bispecific antibodies first, vice versa? And in what timeline? Similarly, can we use one bispecific right after the other? Can we use one that targets BCMA and another one that targets BCMA if one shouldn't work, or another one that targets a different target such as GPRC5D? How frequently should we be dosing? So all of the studies, as I mentioned, primarily looked at dosing these therapies until progression. However, given these therapies are achieving deeper levels of control very early, do we need to continually dose these medications the way we do as maintenance for other myeloma therapies? Or can we help prevent certain immune exhaustion factors that we see by limiting dosing or dosing frequency? And the main point that we want to make here is, in looking at various T-cell engaging therapies, are we creating an environment in which the immune system doesn't have the ability to respond to similar therapies in the future? And that's what we sometimes refer to as T-cell exhaustion, although I'm sure we can have another hour-long discussion on whether we should be using the term exhaustion or not, or a lack of activation, or a lack of a optimized immune environment. However, that being said, are we modifying the immune milieu to such an extent that it may be detrimental to further immune therapy options in the future? I should mention that the reason Usain Bolt is there is that there was a robotics Olympics recently where a robot also beat Usain Bolt. So there's always something coming to beat us. And the same is true in myeloma. I believe the other robot crashed, however. That being said, that brings us to our antibody drug conjugates, right? Do we have other options that may not affect the immune milieu to the same extent? And that led to the development of belantamab mafodotin. As I mentioned, it is an antibody drug conjugate that serves two roles. It serves by utilizing antibody drug cytotoxicity, similar to what we see with the CD38 monoclonal antibodies, daratumumab and isatuximab, however, also carries a chemotherapy payload. It was first approved in 2020. However, in the DREAMM-3 study, it did not show an improvement over as a single agent over pomalidomide and dexamethasone. However, subsequently has been studied in two other phase 3 studies. What I'll focus on here is DREAMM-7, looking at belantamab mafodotin in combination with Velcade versus Dara-Velcade dex, and has also been studied as in combination with pomalidomide in DREAMM-8, compared to a control arm of pomalidomide, Velcade, and dex. And important to note here that both these studies showed an improvement in progression-free survival in patients receiving the belantamab-containing arm, as well as overall survival, as you can see here, which is encouraging for a separate modality in which we can treat multiple myeloma. Now, being an antibody drug conjugate, it does have its own side effect profile. And the most common that we talk about outside of hematologic toxicities, which we do see with both these drug regimens, there was an increased risk of low platelet counts with belantamab mafodotin. However, most notably, there is, particular to this drug class, an increased risk of ocular or vision issues. And so that most commonly presents as blurred vision, irritation, sometimes a sensation of an object in the eye or a foreign body. And this led to the understanding that those therapies, the antibody drug conjugates, based on the chemotherapy that they attach to it, can affect certain areas of the eye and can cause issues with the covering the lens or the of the eye. And so with these therapies, they require frequent monitoring prior to each dose with a ophthalmology colleague that can evaluate for the formation of these microcysts that you see here beneath the lens of the eye, or keratopathy as we refer to it. And in up to a little over a third of patients on the original study led to a decrease in their visual acuity. However, 98% of those patients with a decrease from a baseline of 20/20 vision or 20/25 vision to 20/50 vision recovered with holding the dose of the belantamab mafodotin. And in patients that had a greater decrease in their visual acuity to 2200, all of those patients recovered their visual acuity. So although it is a concern, it is something that can be managed and requires close collaboration with with our ophthalmology colleagues prior to treatment. Now, how can we use these therapies to our advantage? And Dr. Costa mentioned this, that we do see in patients that received by a CAR T-cell therapy, that those patients that received a prior BCMA targeting therapy did have a decrease in response rate and survival outcomes with prior BCMA therapy. Similarly, if we look at those that were treated with bispecific antibodies, in those that had received prior antibody drug conjugates and or CAR T-cell therapy or both, it can affect response rates that we see with the bispecific antibody moving forward. Now, there are several studies that looked at can we sequence various BCMA binding bispecific antibodies, and there are slight differences in the area of the BCMA antigen that are bound, and so progressing on one may not indicate that one cannot get a BCMA bispecific antibody in the future. However, there's several studies looking into can we better identify whether patients have lost both copies of that BCMA binding epitope or just one, and may benefit from a sequential BCMA binding therapy in the future. Similarly, can we use these therapies prior to CAR T as a bridging therapy, like Dr. Costa mentioned? And what we have seen, as you can see in the bottom right here, that patients that received a separate target, so a therapy such as talquetamab that targets GPRC5D, outcomes were improved with CAR T-cell therapy, as opposed to those that received a therapy targeting BCMA, or therapies that included alkylators or chemotherapy such as cyclophosphamide that we use in lymphodepletion, as it may change the immune effector T cells in the bone marrow and the peripheral blood. So, what are other ways to maintaining the immune system to optimize the response to either bispecific antibodies or antibody drug conjugates? One is looking at decreasing dosing frequency earlier during therapy and maybe not waiting till 6 months at the time of response. And this is something that we have based on several real-world studies and analyses that looked at patients that had stopped these therapies because of a toxicity or an infectious complication, but have since sustained a response regardless of stopping these therapies for prolonged durations. Similarly, can we augment the exhaustion or the lack of immune innovation or intervention by utilizing therapies that may energize or reinvigorate the immune system? And that one example of that is shown here with the use of iberdomide, our recently approved CELMoD agent that Dr. Mikhael will touch on in a moment, that may create an environment that can improve the T-cell milieu going into these therapies. And lastly, can we use fixed duration dosing regardless of response to help maintain a healthy immune environment? So where do we stand currently? Bispecific antibodies and antibody drug conjugates offer durable off-the-shelf options that can be used alone or in combination with other therapies in early relapse, in many cases approved now after 1 to 3 prior lines, or in the case of other bispecific antibodies after 4 prior lines. That being said, the toxicity profiles can vary depending on the drug class and should be used to weigh against a patient's personal goals or preferences when deciding on what therapy to move to next. The evolution in therapy for multiple myeloma includes improving patient experience and accessibility by moving these therapies from step-up dosing in the inpatient setting to outpatient, reducing the frequency of dosing or limiting the time on therapy, minimizing toxicity by using medications such as tocilizumab to prevent cytokine release syndrome or minimize the grade seen, and then allowing for the use of combination regimens to improve response rates, especially in patients with high risk or aggressive behaving disease. And lastly, utilizing similar targets sequential is possible. However, understanding how best to use them to maintain immune activity or limit antigen loss and immune exhaustion remain to be studied. Thank you very much for your time.
Great. Thank you so much. All right. Wow. Lots to take in there. Lots of great information. Thank you so much, both of you. We're running a little bit behind time, about 15 minutes behind, but we're going to catch up some time here. We're going to take a little bit of time for Q&A. Just real quick, because I know it came through to me personally. When Dr. Midha says off the shelf, she means that we don't have to wait and collect and manufacture something, that literally we take a drug off the shelf and immediately give it to a patient, as opposed to in CAR T, when Dr. Costa was explaining the whole process of CAR T-cell therapy, we don't think of that as off the shelf because it takes a little bit of time. obviously for us to collect the T cells, to have that 4 to 6 weeks of manufacturing, and then give them back to patients. So great talks. Wow, there is so much Q&A here. We have over 50 questions that have come in. We're going to do a little rapid fire question here. Someone has asked 2 questions about AL amyloidosis. I think for time's sake, I'm going to have to defer these. But apart from saying that AL amyloidosis is kind of a cousin to multiple myeloma, a lot of the same therapies we use are being used in AL. They're not yet formally approved for CAR T bispecifics or antibody drug conjugates, but being investigated. Someone asks, is it efficacious to use DARA with another drug at relapse if DARA was used in the last line of treatment? I couldn't have planted a better question. We want this question because we actually quite strongly believe that the answer is no. Meaning, if someone is on, let's say, daratumumab plus Kyprolis plus Dex, and their disease breaks through, we would typically not recommend keeping the DARA going and switching out the Kyprolis for pomalidomide. We usually like to see a change. Here's a question, Luciano, I'll direct it to you. When you relapse, is it good to go back to the same meds you were on before?
I would say almost never. There was a time and a day where we did a lot of that, trying to extract the very last drop of ever treatment. Nowadays, fortunately, that's not longer necessary. So with almost no exception, once the myeloma relapse, there's always something better to go to that you don't have to go back to the prior drugs.
Agreed. I think the evidence is clear that it even though it may work, it typically doesn't work for as long as it did the first time. And while I have you, Dr. Costa, let me ask you this one, because I know this is an area you've explicitly studied. I've been MRD negative for the past several years. My recent bone marrow test showed that I am now MRD positive. All other labs were the same over the past few years. Does this change in MRD status indicate relapse?
Great question. And that's one of the most controversial things we have in modern myeloma therapy. And I would say the data shows that people who become MRD positive, in general, they go on to progress after several months. That may be 20 months with prior therapies. More like 10 months where people are progressing after more recent therapies because there are better therapies. Progression tends to be a little bit faster. And there are clinical trials going on right now studying whether to intervene just at the MRD resurgence, as we call, or to wait for traditional IMWG-defined progression. And I would say the current standard is not to treat yet just for MRD resurgence, but every case is different.
Yeah, I think you captured it. I think the essence, the concept, and this brings us to another question here that was asked. Someone saying, their blood tests have been good. They were transplanted 3 years ago, but now there's a little trend up in their kappa light chains, but the ratio is actually not that high. The absolute, the ratio is 5.6, for example. And someone's asking, what do I need to do? What tests do we need to do? The way I think about this is, the biology of myeloma is different in every patient. Some people are very, very slow to relapse. Some people, unfortunately, relapse very quickly. And so it's hard to know because these are not like measuring, let's say, a lung tumor where you can really see it on an X-ray and you can tell how big it is. These numbers in the blood, they can fluctuate. They can go up and down. And we've seen patients, like you said, Dr. Costa, who go MRD positive and then jump back to MRD negative. Somehow the immune system caught those few cells and brought them back. So for this person asking about the light chain, similarly, I think we really need to see a consistent trend upward. And as we see that trend upward, then we repeat the bone marrow, we repeat the PET scan, try to get the full picture. Some of you heard me say, I think of myeloma as a crime scene. One piece of evidence doesn't tell you the whole story. You need multiple pieces of evidence, often x-rays, blood tests, even urine tests, to put it all together. Someone's asking, who makes these therapies? Well, we have lots of different therapies tonight. There were some made by the companies that were on the list and others. So multiple. We won't go through every one of them, but we have multiple companies. In fact, even more coming to the fray as there's more and more options for treatment. Dr. Midha, I'm going to ask you this question. Oncologists seem reluctant to do a second stem cell transplant at relapse. Why do you think that is?
So stem cell transplant, I always tell patients, is really giving patients a high dose of chemotherapy and rescuing the bone marrow with stem cells. The stem cells themselves don't treat the multiple myeloma. It's the chemotherapy that's doing the heavy lifting there. And we've had data from various transplant studies looking at the utility of transplant in deferred relapse and or a second transplant at the time of relapse that doesn't tend to show a significantly improved survival benefit in many cases, and comes with an increased risk, especially of long-term toxicity, including a detriment to quality of life, given the immune suppression and infection risk immediately following transplant, as well as the risk of secondary malignancies over time.
Yeah, so I think you're right. I mean, I think the days of second transplant are much, much less than they used to be with the great options we have. I'll take another couple of quick questions, and we're going to take a quick break. Someone said, did the clinical trials for iberdomide include people previously exposed to and refractory to DARA? The quick answer is no. They couldn't have been refractory to DARA because both arms contain daratumab. That wouldn't have been sort of ethical to do. So it is primarily looking at a group of people that are not currently refractory to DARA. But I would say I'd be comfortable treating someone if they had daratumab, let's say in their induction before transplant, and they had never been refractory to it. I'm on lenalidomide and I have a bad itch. Would it be better to try Revlimid? So, this is now getting into the days of generic uses of drugs. This is something to speak to your healthcare team about. There are some patients who will actually have a bit of a rash and itchiness to one of the compounds and not the other. So sometimes that can be a switch. But I would say that most of our patients that develop itchiness or rash with any of the compounds can actually overcome it if we are careful with how we manage it. We typically give steroids for a while, stop it, restart it again, sometimes even a bit of a lower dose. Most people can stay on it. Lastly, I'm going to ask Dr. Costa this last question. Has anyone considered using harvested stem cells from an old transplant collection to replenish T cells after failing immunotherapy?
Not in a formal way, not in the setting of like a prospective study. But I would be lying to you if you said we haven't done this. Sometimes we worry about the patient's, patient has, low blood counts after too many therapies. They have received several therapies that affect the function of the T cells. So sometimes we, do a boost. We just infuse cells, knowing that they will help improve the blood counts, but also at least theoretically improve the health of the T cells, so patients can subsequently yield a CAR T product for their own treatment, or they can have a better response with T cell engagers. But it's something that is very hard to study prospectively, so there's not a categorical answer to that.
Great. Excellent. Well, I wish I could get into all these questions. Maybe I'll just take one more because it's so, or two are kind of lumped together here. Someone's saying that they're progressing on DARA and POM. Should they go to Kyprolis and DARA? And I think we've kind of answered that already, that's typically not what we would do. It may be worth having the conversation about either a CAR T or the bispecifics that Dr. Midha just discussed. And let's end with this question, because this is a very positive note to go out on a break on. Is there any new data yet on the trials where CAR T is used in earlier lines of therapy or first, second relapse that are showing better results than when we used it in later lines? Well, actually, Dr. Costa showed that, didn't he? He said, no, based on the number of lines of therapy, patients did better earlier. And with the CARTITUDE-1 results, with the exciting news that 30% of patients were without progression in 5 years, was there ever a comparison that those stats would be for those patients, a similar cohort used earlier? And the quick answer is yes, we are studying that. We are looking at that. One of the reasons why we haven't had the long-term results of the earlier line therapy of CARTITUDE is that those patients are still doing really well. So I think the essence that Dr. Costa and Dr. Midha were sharing with us is that when we bring these treatments that initially get approved after multiple lines of therapy early on, patients do better, typically have a deeper and a more durable response. And Tricia, after the break, is going to explain to us a little bit more. That both of our speakers talked a little bit about some side effect management, but she's going to talk to us a little bit more about that because, interestingly, sometimes when we use these therapies in earlier line, we also actually have less side effects for various reasons. So we're going to walk through that when you come back. That's your teaser, because we're now going to take a quick break. We're running a little bit behind, but we're not too bad. We're going to start sharp at 1:45, whatever time zone you're in. So that's about 6 minutes from now. So maybe we can set a quick timer for about 6 minutes. We'll take a break. And then when we come back, Trisha is going to talk to us about how we can manage the side effects from these great therapies. So everybody can take a quick break. We'll see you back in 5.5 minutes. Great. Well, welcome back, everybody. I hope everybody got up and moved around. As I say, as a hematologist, to be professionally embarrassing if anyone gets a DVT. So we want everybody up, moving around. And now we have 45 minutes left in our webinar, and I am thrilled to invite a friend and a dear colleague on the IMF Nurse Leadership Board. Tricia Mangan is going to come and speak to us about putting this all together, especially around the supportive care aspects, as we call it, or side effect management of these great therapies we've been talking about. So the floor is yours, my friend.
Oh, great. Thank you, Dr. Joe. And so happy to be here tonight. The focus of this discussion will mainly be managing some of those toxicities that we highlighted in the last two discussions. But before we get there, as just to remind us of just with the disease itself, we are talking about relapsed disease, as Dr. Joe went through. We know that ultimately we have great treatments at every line of therapy. We have great expectations that you will respond and drive that disease into remission. But ultimately, over time, many of our patients, their disease does start to progress. And there's a lot of decision making throughout this process. And what I really want to highlight is really how we partner together in over time, we become not only a relationship with a patient and providers, but we become friends, and we really develop a trusting relationship over time. And that's one of the most beautiful things that I've experienced in all these years doing this, that, we didn't know many of our myeloma patients years ago. I watch them celebrate graduations, weddings, birth of children. So we look forward to this. And through this process, we do come up with time points where we have to make some critical decisions as the disease might progress. And so when that time of deciding when treatment should change, it does, as Dr. Joe mentioned early on, that there are some disease-related factors. How quickly is your disease progressing? Is it something that we can watch and monitor? Is it growing? Are the numbers changing much more aggressively? Are you developing more bone lesions than you had in the past? So there's disease-specific changes that may occur, but there's also other issues related to how you're feeling. Do you have other comorbid or other conditions that may play into our decision making as well? So we're always looking, as many of your questions had asked, if I've had this treatment in the past, what treatment should I go towards? And so with that time and relationship, a discussion occurs. And we definitely, hopefully, we kind of understand your preference, but we need to hear it again and again, because your goals change over time. Where you might have been working and I want oral therapy, but now I'm retired. I have more time to come to the office, or things like that, or vice versa. So I think, we definitely have a lot of options of therapy, as you've seen, and hopefully are walking away from this discussion with that it's exciting time. And we do value, and it's very important for not only your preference, but your support system as well. Let me just, or did I hit too many times? No, I'm sorry. It is getting late, sorry. So shared decision-making is the term that we use, and it really is that discussion that not only do we have to discuss where your disease is at right now, what are the options of therapy, what are your goals and preferences? After you hear, we lay out all the options. And as you all know, the waiting room can be long, that wait, as every patient is having these discussions. And so, really hearing what you have to say makes this more important, and hopefully will be better in terms of outcomes because of your involvement in the decision making. So we want you to ask questions. In fact, prepare for them, write questions down. You may have heard things from support groups or with the IMF, other patients that have gone through different things. We want to, jot down this, all the questions that you might have, just so that at that discussion, you are able to make sure you touch on all your issues and concerns. We also like to know you should listen to what your providers think, knowing you, understanding where you have been, what types of therapies you've had, what is their suggestion is always a helpful thing. But I like this last bullet point on the right-hand side here. It's like, sometimes we are so used to having these discussions, and this is like, we do this all day long. For you, this is the first time you might be experiencing a relapse, or, here we go again. What do I have to do? The answer is take your time. If you can't make a decision right away, say, wait a minute, let's figure this out. Let me go home, discuss it a little more. One important thing is sometimes if the relapse is somewhat aggressive, or even at diagnosis, some people present a little more aggressively than those that may have a mild presentation. Sometimes we do need to kind of make that decision a little quicker to get you well again as quickly as we can. But we do appreciate you should ask for additional time or maybe further clarification to make this decision. And just to make sure you understand the options, that you feel comfortable, not only you, but your support staff really understand and feel that this is a good decision for you. And we I can't speak highly enough of a lot of this work. As Dr. Joe said, our nurse leadership board really got this out there to other providers and nurses and advanced practice nurses for this discussion, just so patients are aware. I love these little tip sheets that the IMF have put together, just highlighting some of the questions that you might not even be considering, but just having access to asking your doctor questions, how to set that up, or just to be more knowledgeable about that discussion if you're uncomfortable with that. But I really like these tools, but also you are your own person. You've been through it. You know how you feel. Write these things down. The only way we know is if you share it with us. What are your concerns? What are your preferences? Those types of things. So we welcome this discussion, and we look forward to this discussion because we know it'll be successful when we do it. All right, that was shared decision making. And now we're going to move on to all these exciting immunotherapies that we've just highlighted at Relapse. There are some that many have some side effects, and I really wanted to spend some time helping us all understand how we manage them. And so not only from the treatment itself, but the disease itself, especially at relapse. Some people have a newer presentation. Some people might have a new bone lesion, or as Dr. Midha had mentioned about lumps of plasma cells or plasmacytoma, extramedullary disease, we call that. That might be new. You've never had one of them before. So having an understanding of the disease itself growing can cause side effects. So elevation in calcium as the disease is growing. Some people can have some renal insufficiency. Certainly low blood counts is something that we are always monitoring for, as you know, and with that comes the risk of infection. So if you have pain or neuropathy, nerve pain, things like that, and we'll highlight fatigue. So we will touch on all of these causes of some symptoms that you may have, but I will also touch on the treatments, what can happen from the treatments, GI symptoms, low blood counts, risk of infection. Blood clots can happen, as Dr. Joe said. We have to get up and move around. But the other thing is, when we took our break, we should drank a glass of water just to stay well hydrated as well. That's always an important thing. So let me move on to, again, the immunotherapies that are so exciting. We know that our immune system is so vital in breaking, really combating cancer or abnormal cell development. And so we have CAR T cells is very quickly that are genetically modified. You take out your own T cells, you genetically change them outside of the body to put a new, what we call like a T cell receptor or warhead that has great specificity to a particular protein glob on the surface of myeloma or an antigen. As we know, all of the CAR T cells target BCMA antigen on the surface of the T cell. And there's two active, one soon to be approved by the FDA, hopefully maybe by the end of this year. We know that process of one person identified as a potential CAR T patient. We collect their T cells in an apheresis-like procedure. If you've ever had a auto transplant, you know we harvest your stem cells through apheresis, and this is a machine that just gathers your T cells. We then send them to a manufacturing site to be manufactured, and that takes a few weeks to do. During that time is we're trying to keep your disease in check, and I think this is an important aspect because the better your disease is controlled at the time of your T cell infusion, we do think that minimizes some of the potential toxicities that you may have. And so bridging therapy is a very important aspect. It's different, as Dr. Costa said, for everyone, depending on how aggressively your disease is growing and such. Once the cells are manufactured, we get called from the manufacturing team. It is good to go. We then plan that lymphodepleting chemotherapy to clear out the other lymphocytes in the body so that when those CAR T cells are infused back in, they have no competition. They can very aggressively grab onto those myeloma cells and proliferate and expand in the body. And so we monitor closely for those potential toxicity, which is the next slide we'll talk about. And then eventually you will, we tend to do that either inpatient for the first week or longer, depending if you have a side effect. But some CAR T centers will even actually have the ability to do it as an outpatient, where you stay very local, come in every day for monitoring, for that close monitoring. And patients are very successful with doing that, even in an outpatient setting. Bispecific or T-cell engaging therapy is, as mentioned a couple of times already, off the shelf. It's already manufactured. It engages the T-cell. So the antibody, one part of the antibody grabs that antigen or protein glob on the surface of the myeloma cell. The other part of that same molecule grabs your T cell and very aggressively brings them very close together, causing a great amount of immune stimulation and destruction of that myeloma cell. And so we like that because it is available. We can identify someone, and within a day or two, we can start that therapy up. And so we have two types of or targets. We have BCMA target, just like the CAR T cell, but we also have this GPRC5D. It's very wordy, but it's just another protein glob on the surface of the myeloma cells. And I'll highlight that there is some different toxicities. Any of you who are on this call, who are on that, you know there are some different side effects that potentially happen in that circumstance. So, but let's talk about the two main toxicities of immunotherapy, because we're engaging those T cells and trying to really more aggressively destroy these myeloma cells growing. So the first syndrome is called CRS, or cytokine release syndrome. It turns out your T cells, when engaged or when you have them genetically modified in the CAR T cells, they really cause immune inflammation. And so cytokines or chemicals that are in our system can become very activated. And in particular, there's a interleukin-6, which is one of the main drivers is thought in the cytokine release syndrome. Well, it turns out we have an antidote for that. It's called tocilizumab. It's an anti-interleukin-6 antibody that can be administered if you experience or were suspicious that you're experiencing this CRS or cytokine release syndrome. The syndrome itself always starts with a fever. And so that's the first symptom that we see. We start monitoring you very closely. In some circumstances, that fever can get pretty high, but sometimes it's a low grade. Sometimes we monitor people that may never need tocilizumab. It's a very low grade, but we're always ready to use it when it comes to CAR T cells. Bispecific antibodies can also have the same reaction. And in some cases, a way of prevention is we actually can give that tocilizumab as a pre-medication before you even get the bispecific. Some centers are using that in that circumstance. Some we watch very closely, and as soon as there's a symptom, we'll initiate that. So it's an important aspect. So we actually have an antidote that can turn this process off. Steroids. We know that some of these toxicities can have we know that it's T cells, which are lymphocytes. And so what do steroids do? They kill lymphocytes. So turning off this process, if you're getting sick from cytokine release syndrome, can be a very helpful thing to do. And that comes with time. You know, when I fortunately have been working with CAR T cells and immunotherapy for over a decade. And there's a learning curve with that. We got very comfortable with, like, we were very afraid to start treating people with side effects. We don't want to turn the process off. We want you to respond to these therapies. But it turns out if you are developing these syndromes, it's more important to turn the process off so it doesn't get a higher grade or get you even sicker. So we're monitoring for that. The other thing that it's really monitoring, giving some of these antidotes, and in bispecifics, let's talk about that, because unlike CAR T, where you get it manufactured and the whole bag of CAR T cells get infused into you in a one-time infusion, bispecific antibody, we do something very important in the beginning to avoid some of these toxicities of cytokine release, where we give a very small dose called step-up dosing, where that first week you either get admitted into the hospital or sometimes close by and come into the outpatient center just to be monitored during that first week. We give a very small dose. We monitor you very closely. If you have a side effect, we're managing that side effect. We will hold off giving you that second step-up dosing, a little higher dose, after 48 hours. And then if we monitor another 48 hours, if everything is good, we'll give you that first full dose. That is what we call step-up dosing, which we can control the amount of these antibodies that we're giving you, and hopefully that can avoid some of the worsening of some of these toxicities. The ICANS, or immune effector cell neurotoxicities is what we call that. It's very common in myeloma that it is a lower grade. It can be serious in some cases, but most people notice a little headache. I work with a doctor who's very scientific, and he says, I don't know, they just feel mucky. And it's like, it's not really, sometimes they're just not feeling well. They're sometimes a little lethargic. Some of these symptoms can occur. And we have learned from, if you start to experience some of these, we'll start to give some of the antidotes, in particular steroids can be very helpful in avoiding some of the worsening of these toxicities. There is also listed here delayed neurotoxicity, which, as Dr. Costa mentioned, sometimes people can develop like a Bell's palsy or a cranial nerve, like some of the nerves that innervate into our face. You can have a little one-sided, like some, a little droop or something like that. And so we're very quick to start making sure everyone is on antiviral prophylaxis is a very helpful thing. Monitoring for this, usually it's transient. It usually is a little more delayed in emerging, but we are monitoring for those things, and we'll, certainly hold off on any further therapies in the bispecifics until some of these symptoms improve. But we have gotten pretty good at managing these toxicities. The other thing I do want to mention that is kind of, I'm sure we all in the practices that we all come from, we look at your lymphocyte count when we start to give these therapies. And if your absolute lymphocyte count starts to increase, we associate sometimes that can increase maybe the likelihood of developing some of these toxicities. And so some centers will give a little steroid for no reason other than your absolute lymphocyte count was elevated. And we'll give a little pulse dexamethasone for a few days just to try to lower that. And hopefully we'll augment, any worsening toxicity later down the road. And again, mentioning earlier in this discussion, trying to have your disease at the best remission we can at the time of starting these therapies is very helpful. But sometimes we can't always get to the most ideal spot, but we try to lower the disease burden, which can be helpful. Infection. So one thing that we do know about these immunotherapies is that over time, your immune system is affected by this. We know myeloma in general, you have a higher risk of infection, but we know when you're on these therapies or have recently received them, we're monitoring very closely for infection. We do, these are just good tips overall that you want to stay well, practice good hygiene. You want to avoid people that are sick. The number of questions I have of like, I really want to go to this wedding. Can I go to this wedding? And the answer is, sure, you have to take risks and benefits to everything that we do. But trying to avoid large crowds or people that are sick, that's a good tip. Washing your hands is probably the biggest thing that we can do. Getting immunizations like the flu vaccine every year, keeping up on the COVID, RSV, and the other types of vaccines are very helpful. But more importantly, when we start these immunotherapies of CAR T bispecifics and antibody drug conjugates, we do usually initiate some different antibiotics to prevent infection as we start these therapies. And many of them will continue for a period of time, particularly the antiviral medications. So these are helpful things just to be aware of with infection. And always taking a fever serious. I'm sorry, I switched that over, but 100.4 is a meaningful temperature. And so we do want to be called about that. These are typical infections that people can have, antiviral prophylaxis, if especially you can reactivate your chickenpox or have cold sores, things like that. Bacterial infections will often give broad spectrum antibiotics if you have a low blood count or you're at a period of time where we're concerned that you might be at a risk for developing some of these other issues. We have you on PCP, or there's certain types of atypical pneumonias that people can develop. And so Bactrim, you may have heard that and maybe on it. We usually have people on that Monday, Wednesday, Friday, something like that, if they can tolerate it. Fungal medications. One of the therapies, teclistamab, was done, that study was done in the setting of right peak COVID. And you'll see when you saw some of the results from Dr. Midha that the COVID infections was higher there, and it was because of when the study was being done. But what we learned from that so much, we learned that being on prophylactic antibiotics can, from the beginning, is very helpful to prevent various infections, not only COVID, but also other, certainly other infections that may develop. One thing we monitor, and if you are, we always monitor your quantitative immunoglobulins, your IgA, M, and G, but it turns out your IgG level can become very suppressed with some of these immunotherapies. And so we're always monitoring that and initiating IVIG, infusions of IgG, which we find very helpful to maintain a good humoral immune level to avoid infection. So many of you are probably on that, or I don't know, I think we draw more IgG levels and initiate that therapy. And it really can be very helpful to avoid infection. And monitoring blood counts. I mean, we know that if your ANC, or the absolute neutrophil count, dips below 1,000, you're at a higher risk of developing infections. So initiating some growth factors to stimulate your white count to create those neutrophils to help your body fight infection is very helpful as well. So these are some highlighted tips for infection risk. One other toxicity that we talk about with that talquetamab, that GPRC5D target, as Dr. Midha had mentioned, it's not only expressed on the surface of the myeloma cell, it's also on nail hardening cells and and skin. So you'll see these are some of the changes in people that do take this medication. It can cause some dry skin in feet, hands, the nails. You can see that they can have changes there as well. These changes occur over time, usually early with the initiation of this therapy. And so we're very quick to lather up with creams and keeping your nail short. Some people find that as the nail can sometimes pull away from the nail bed, putting a Band-Aid on it, things like that we have found helpful. Moisturizing with Vaseline can be helpful as well. These improve over time. I want to assure you of that. And so we can always hold the therapy or extend the therapy longer if some of these toxicities occur. And then the other toxicities that were mentioned earlier, dry mouth. Some people have altered taste or loss of taste. And so it can be somewhat of a challenging side effect, also associated with that talquetamab or GPRC5D target. And so we are very quick to, it doesn't happen to everyone. We prepared one guy who was like, okay, and he went and gained 10 pounds before he started the therapy. And he didn't have any of this difficulty tasting anything, and he's like, now I have to lose 10 pounds, but it's every treatment is different for different people, but these are common things that may develop at varying degrees. So we do find some people have difficulty swallowing. There's a dryness or weight loss. We do encourage you to, we sometimes partner with our nutritionists and things like that. We're very, I like some of the suggestions already with some liquid saliva, just staying liquid foods, soups, things like that. People like a little better. Bread can be somewhat challenging because it's dry and you have to drink some fluid while you're eating these things. But these are things that we work with together, and letting us know that you're having these issues is part of the thing. This whole communication is so important, that we know what you're feeling so that we can help with these toxicities if they are developing. And it doesn't, some people are afraid to share their side effects because they know the treatment's working and they don't want us to stop it. That's not the right answer, because we can spread out the schedule of things. We can help you manage these toxicities better. And over time, many of these do improve. GI issues are not only associated with some of the immunotherapies, but also with the just other therapies that you've been on. And so constipation, we know, can be related to treatments we give, but also pain medicines we give or other supplements that you are on. So staying well hydrated, trying to eat high fiber foods may be very helpful. And some people do require a bowel regimen to really stay on and staying well, having normal bowel movements. Anorexia is certainly a frustrating thing. As mentioned here, it can be associated after transplant, or some people that are on that new targeted GPRC5D target can have some of these struggles with appetite and things. So small, frequent meals, I do like that. I think people find that very helpful. And maybe more foods that are softer or liquid can be very tasteful. I always find thing that I never think of, I love ice cream, but sherbet, it turns out that sherbet that have some of those more stronger fruity flavors can break through some of this loss of appetite, and it's also a liquid. So a lot of our patients find that those wouldn't really think about a sherbet, frozen sherbet, but can be very helpful. In Philadelphia, we call it water ice. So, but that's what we would encourage as a way of getting some liquid in. Diarrhea can certainly be associated with many of the things that we're giving. Antidiarrheal medications can be very helpful. Ruling out an infectious reason for diarrhea. Many people may have a higher risk of developing a C. diff colitis or something like that, which is an infectious thing that we can give an antibiotic to treat. But staying well hydrated and letting us know if this is happening. I had a woman come in, sometimes, just the way someone walks into the office, you're like, "You've lost weight. What are you doing?" She goes, "But this is the first time I've gotten into my jeans from high school in the longest time." And we're like, "Well, that's good, but we want you to be healthy too. So please let us know, and we can work on certain approaches to dealing with the GI toxicities." Now, there's other side effects that are associated not just with relapse, but throughout the disease trajectory of myeloma. Fatigue is the most common complaint people have, and it's a very common thing. And so but let us know if it really is becoming a difficult thing, because many people say, it's just part of the disease or something. But maybe we can help with dose reduction or adjusting of certain medications can give you more energy. Staying more active can be helpful. Anxiety, understandably so. It is not uncommon that people can have some anxiety, especially if their disease is starting to grow. What type of therapy am I going to go on? What will happen? And certainly depression can occur too. So we realize this can happen. We want to have that discussion. There's things that we can do to help you with that, with counseling and just and things that we find people will benefit from. So letting us know is certainly a very important aspect to this. pain management. We know that pain can come from disease itself. If you have a painful bone lesion or a fracture of something, or if you have neuropathy or some compression fracture that's pressing on a nerve in your spine that's wrapping around or something, we do find that pain can be an issue in our patients. Also listed here is shingles, that we call it post-herpetic neuralgia or pain syndrome after a shingles outbreak. And that's why it's important to take the medications that we prescribe in case of that reactivation of your chickenpox to prevent shingles. There are medications that we can prescribe. There's other therapies that we can do for pain. If you have a painful bone lesion, radiation therapy, the doctor I work with always says a little spot welding to an area might be very helpful for pain management of that, as well as controlling the disease that's causing that, as well as other procedures. Getting physical therapy involved can be very helpful. Many patients do find yoga, acupuncture, other alternative therapies can be helpful, along with other traditional therapies in managing the pain issues that may develop. So it's just something to be aware of, these different options. And then just finally, I wanted to touch on the kidney, just staying well, bone and kidney health, staying well hydrated. I want to get to the questions, so I don't want to spend too much time, but one of the biggest parts of all this is to have, we expect you to be hanging around with us for decades, and we want you to keep up on your other health, see your PCP, get your mammogram, get your colonoscopy, see your dermatologist. Those things can be very helpful in quality of health and long-term health. And certainly using, involving your care team and getting help for the more social aspects you have can be very helpful with your anxiety and other things. So we do want your family and you to have a normal life with all the quality that comes with it. And this is nice little tips on your care partners to help with helping you and themselves as well through this journey that we have. And I think I will these are just all the teaching tools that are put together by the IMF. And so I'll just end with that. And this is just our IMF Nurse Leadership Board. I was actually not present in that picture, but a great group of nurses. I'll end there.
Great. Well, thank you so much, Tricia. I know that wow, all three of you have covered a lot of material. So this is fantastic. Really, really helpful, practical things. A crazy number of questions. I'm going to go to some of the questions before I wrap up with a little bit of looking into the future. Lots of great questions here. I just want to address this one because someone obviously may have a concern. I understand it. Are any of the treatments not from the sponsors? Let me be very clear. I have no idea who the sponsors are when I put together these slides. I find out on the day of, and we've spoken about every myeloma therapy independent of the sponsor. So just to be very clear, the sponsors don't influence what we discussed tonight. We're grateful for them, but there are many, many therapies discussed tonight that are not represented from the sponsor list. So please be reassured that the IMF really takes conflict of interest seriously, does our very best to ensure that we present everything in a balanced manner. We're also excited about all of these new therapies. It's hard to really associate it with just one of them. Luciano, maybe I'll ask you this question. Do you believe anita-cel will have advantages over our current 2 CAR T-cell therapies approved? I was going to show that in the future of Myeloma Talk, which I may show in a minute, but just quick thought on anita-cel?
Yeah, I think there's a lot of optimism and hope for this new CAR T for some reasons that I'm sure you're gonna show. But what I want to warn people is this CAR T is likely to be approved at the end of the year. based on single-arm trial for patients with more advanced myeloma. So we're going to be comparing apples and oranges, and I think we're going to have to wait to see how that treatment performs in earlier lines to start establish some sort of cross comparison between that and the other existing CAR-Ts that we have. But I think we're still going to see the data that supports this approval. We haven't seen the entirety of the data, but we're not going to have a clear answer at the beginning. But in a way, it's good that we have more choices, and you're going to be able to sit with your physician and analyze the characteristics and the approvals for each CAR-T to choose which one is best for you.
Excellent, excellent. Someone asks here, Dr. Midha, is Blenrep or belantamab mafodotin a good idea right before CAR T-cell therapy? Is that a good holding therapy before CAR T-cell therapy?
Yeah, so that's a great question. I think we do see that sequencing BCMA therapies prior to a BCMA CAR T-cell therapy tends to lead to a decrease in the response rate. However, it's a little bit less so with the antibody drug conjugates than with the bispecific antibodies in some cases. And then, it's something that can be taken into account if it is feasible for the patient. I've also seen good success anecdotally with antibody drug conjugates following CAR T-cell therapy. So always one to keep in mind in that respect as well.
Excellent. Thank you so much. Tricia, if someone has chronic kidney disease, is a bispecific medication still available to them?
Yes. Yeah, I think it really doesn't seem to impact that. We've successfully been able to treat people with bispecific therapy.
Excellent. Someone asked, can an individual with grade 2 keratopathy receive belantamab? This individual is in relapse. So the quick answer is typically no. We want it to go down to grade 1 or less. So as Dr. Midha pointed out, when we give belantamab mafodotin, this drug can cause a little cyst. And, my brother would be very proud because he's an ophthalmologist. I really don't know the eye that well, but there are 5 layers of what we call epithelium, or if you will, skin of the cornea, the outside of the eye. And the belantamab mafodotin issue is only in that 5th outside layer. And so it just gets shed, and then the issues go away. But we want to make sure that it's gone down to grade 1 or less, typically. And then I saw a question here towards the end that I really wanted to ask Tricia. When I talk to my oncologist about fatigue, he tells me that I must remember that I am 77. But I have considerable fatigue during my 21 days on Revlimid, which reduces in my 7-day break. He says that I can't stop the Revlimid because I'm at high risk. Do you have any thoughts or hope there?
Well, I think certainly the dose is certainly an important thing initially, but over time with maintenance, it's just the act of being on it. So this is a great example of dose reduction can really help with the toxicity that you might be having and maintain the remission that you're on while you're on the Revlimid. So I think that is probably one of the main reasons, that and some often diarrhea long-term on IMiDs, particularly lenalidomide, to really dose reduce is fatigue and GI toxicity.
And we hope, I think, too, a time with the cell mods that are similar to them with the iberdomide and zigdomide, that we're actually seeing less of the fatigue, less of the diarrhea. I'll take one more question here. Someone says, does giving toci or tocilizumab lessen the efficacy of CAR T or bispecific? Has this been studied? And I think we do know this, that it really does not. It does not reduce them. And I think with time, I don't want to be too much of a prophet, although I am going to give you a uber fast lecture on the future of myeloma, that I think we're going to be using a lot more TOSI, to be honest, because I think we're learning that if it keeps people out of the hospital, if it reduces the rates of cytokine release syndrome, it can really make a difference. So looking to the future after all of this discussion, we've talked about CAR T-cell therapy and the 2 CAR Ts that we have now with ide-cel and cilta-cel. But looking to the future, we have all these new things coming in CAR T-cell therapy. Let me walk you through them, looking at different targets, making them faster, so-called fast CARs, binding the CAR T, binding the myeloma cell more effectively. That's one of the very important things. When Dr. Costa had mentioned we may sometimes see these side effects, and Tricia even mentioned that we may be looking at people's lymphocyte count, which may be reflective of their CAR Ts. If it goes up too much, what we're worried is that sometimes these CAR T cells, these are like amped up soldiers ready to kill myeloma. If there are too many of them or they're growing too quickly, they may traffic to places that we don't want them to traffic to. So if we can make sure that they're only going to hit their target, no friendly fire and going elsewhere, then that could be exciting. And then there's the in vivo and allo as mentioned. So we have, for example, a CAR T that doesn't just bind BCMA, but actually has a dual target, binds BCMA and something called CD19, which hopefully will allow it to more carefully bind the myeloma cell. We have the anita-cel that Dr. Costa answered the question about, where here it's not dual targeting, but it's something called D-domain binding, where again, the idea is to, in a more enhanced way, target the myeloma cell. Then we have this beautiful concept of in vivo CAR T, which, right now we call it ex vivo because I take cells out of a patient, I manufacture them, I give them back to a patient. What if I just gave them a drug and trained some of their own T cells to become CAR T cells? This is not a science fiction movie. This is actually what we're doing, and it's really remarkable. And I know it's early, but the first 18 patients treated with this, all 18 went to MRD negativity in the first month. So it's quite exciting. And then Dr. Costa also mentioned this notion of allo CAR T. Could we collect T cells from healthy donors and manufacture CAR Ts and then just give them off the shelf? So you get kind of the best of both worlds. You get the best of Costa and Midha. You get the best of a CAR T and a bispecific together, right? But the joking aside, this is something actually I'm just opening a new clinical trial this coming few weeks with a brand new allo CAR T that has been developed. Of course, we heard beautifully tonight from Dr. Midha these wonderful bispecific antibodies, in particular those that target BCMA, but now are targeting GPRC5D. Well, what's the future? The future is going to be yet another target called FcRH5, using drugs less frequently, having less cytokine release syndrome. And lastly, this whole notion of tri-specific. So first of all, in development, we have a drug called cevostamab, which now has a whole new target. So if part of the challenge with these drugs is that the disease is smart enough and it figures out not to have a target to let the bispecific grab onto it, then now we have to grab onto a different target. And so cevostamab is studying FcRH5. This is a drug called etentamig, which I know there's a lot of data here, but just look at the last number on the screen. When they gave this drug from the start with only one step up once a month with prophylactic tocilizumab or preventative toci, they had 0% cytokine release syndrome. This is going to make it easier and easier. If someone asks the question, do you have to go to an academic center to get a bispecific antibody? Drugs like this will be more easily given in the community. And then all these tri-specifics. What's a tri-specific? If I have two hands, I don't have a third hand. Maybe if I get my leg up here, but try a good yoga pose. But if you have two hands now. Right now, we have one arm grabs on the myeloma, one arm grabs onto the T cell. Now with three arms, two grab onto the myeloma and one grabs onto the T cell. So it allows more effective binding to the myeloma. And in fact, the analogy I give is it's like driving a car. You can drive with one hand. it's better to drive with two. You have a better control of the steering wheel. These drugs are designed with two to more effectively hold on with two targets at the same time to the multiple myeloma cell. And we're going to see drugs like elranatamab and others really come through in the near future. I showed you this slide earlier. It's amazing. I'm just literally covering the surface level. There are all these other new molecules that are coming, different kinds of way of attacking from the outside and the inside of multiple myeloma. These are really, really exciting times in multiple myeloma. I'm sorry that we didn't get to all 67 questions. We got to about 25 of them. I'll try to answer others, and please keep sending in your questions to the IMF. Go use any social media that we have with the hashtag #AskTheIMF, and we'll try and answer your question. As we've noted, you can go to the IMF website and replay this video in the publications and videos section is where you'll find it. Please provide feedback to us. We'd like to know, is this the right length of time? Is this the right time of day? What other topics do you want to cover? Those kinds of things will be very helpful to us. We want to thank our sponsors again: AbbVie, GSK, Amgen, Johnson & Johnson, Johnson & Johnson, Innovative Medicine, Legend, Karyopharm, Kite, and Pfizer. And with that, I am going to take a quick moment to thank our wonderful speakers tonight. I want to thank our audience for listening, even though we went a few minutes over time. We hope this was helpful to you. We trust that this will help you through your experience with multiple myeloma, both you and your team that are taking care of you. Please reach out to us. And again, let me just thank each of you in turn, to Luciano, to Shonali, to Tricia. You are wonderful. Thank you for your kindness tonight. Thanks everybody for joining us, and have a great night.
Thank you.